Good afternoon, everybody. I'm really happy that I've come to this meeting. I've really had a great time. It's so well organized, and I can see all young people interacting and giving talks. Wonderful meeting, and hopefully the last session will be good and you guys can rest afterwards. Anyway, as Sabine said, thank you for your introduction. I'm from the Complex Carbohydrate Research Center. I'm going to tell you a little bit about how we do structural analysis in my lab on different areas of glycoconjugates. This picture actually is a picture of spike protein and the ACE2, but I like it so much that I'm using it. But I'm not going to talk about COVID today, but we have done extensive work in the last 2 years on spike protein and ACE2, so you can read about it. But today I'm just going to give you some examples of some of the work we've done in the last 6-7 months on a couple of our projects. So my lab does structural characterization of glycoconjugates as a whole. So we can start with glycosaminoglycans, we start with tissues or cells, we do structural characterization of glycosaminoglycans using mass spec and NMR. We do extensive amount of glycolipid analysis, a lot of bacterial polysaccharide work, lipopolysaccharide, capsular polysaccharide, and EPS polysaccharides. We do a lot of glycoprotein work from cells or tissues, viruses, including the SARS-CoV-2, a lot of fungal glucan work, and basically we have a bag of tools that we use depending on what is needed. We use mass spec, GCMS, HPLC, MOL-DMS, electrospray, extensive amount of NMR characterization, 1D and 2D NMR characterization, linkage, compositions, molecular weight determinations, capillary electrophoresis. It really depends what the project needs, and we'll use it as needed. So, but for today's talk, I'm only going to concentrate on glycoproteins, and I'm going to concentrate on that side of the slide, basically. Everything on that side is what I'm going to show today. So basically, I'll cover glycomics. And at a later stage, maybe next year, I'll come back and tell you about our glycoproteomics efforts and what we're trying to do for enriching for glycoproteins and glycopeptides prior to analysis. But for glycan analysis, we can start from tissues or cells or singly purified glycoproteins. And the way we usually do it, we do a reduction carboxymethylations of the protein, and then protease digestion, depending on the sequence of the protein. We may do trypsin, or we may do multiple digestions of the protein. And we can release the N-glycans with a combination of either N-glycanase, PNGase F, or Pngase F and A, depending on what the proposed glycans are going to be. There's various techniques that we can use for their analysis. Predominantly in my lab, we permethylate glycans prior to analysis. We also can do labeling of the glycans and then do HPLC or LCMS, but as it may come clear a little bit later, we predominantly permethylate, basically replacing all the hydroxyl groups with methyl groups in order to increase sensitivity for the glycans and also enable us to do detailed structural analysis, sequencing of the glycans, looking at the non-reducing end versus non-reducing end, and the moieties attached to different sides of the molecule. And then we'll do mass spec analysis depending whether it's N-linked or O-linked, and in O-linked, again, we can use beta elimination to release the glycans or hydrogenalysis sometimes. So I'm just going to go through a few examples, as I said, in the last few months that we've done. So basically, this is an example of collaboration with Hudson Freeze and his postdoc Paulina from Stanford Children's Health Research Center. HUD is interested in looking at how fucose is basically what happens to fucose in the cells. Where is it coming from? His question was, he was interested to know the pool of fucose that we have, this homogeneous pool of fucose that we have in the cells. Is it just one big bag of fucose being used for different glycosylation pathways, because we can have fucose coming from exogenous fucose from diet. We can have from salvage pathway. That means it's already been in there. We're going to basically get it from what was already in the cells, right? Salvage pathway. Or we can do, the cells can do de novo sequencing, de novo synthesis of these using either glucose or mannose, for example. So is there one big bag of all these going into one pool and they go to difference, or is there a difference where the fucose ends up depending on how it starts, how it gets to the cells? So what they've done is they basically treated the cells, various cells actually, number of cells they've done, with C13 fucose and wanted to track where it's going in the cells. So the first spectra you see up there is different amount of these C13 fucose starting from 0 micromolar to 50 micromolar. And we can see this is just a snapshot of one glycan, seeing how the ratios change as the external amount of fucose increases, the C13 amount increases, what happens to the molecule, to the N-linked glycan. So what we can see is at the beginning, it's only C12. And as we increase the amount of C13, we can get complete C13 basically. So at about 15 micromolar, we get equal amount of C12 to C13. So the other experiments that was done was looking at the original GDP fucose. How is that produced, and what is the amount of that in the cells? Correlating the amount of that with the amount of N-linked glycans that are present, they were able to deduce and what is the ratio of the fucose from exogenous fucose versus what is coming from endogenous fucose. So looking at the fragmentation, we were able to do MSMS analysis to figure out where the fucose is going to. So when you feed these cells with C13 fucose, the exogenous fucose predominantly goes to the core. So the first place it goes to is the core. And then after that, if there is enough of the C13, then it starts going to the antenna. So, but if you have very limited amount of exogenous fucose coming in the lab, it only goes into the core. So, and then from the MSMS, we can kind of calculate the ratios of what is the amount going to the antenna versus going to the core. So why is that important? Why is he interested in that? Because exogenous amount of these monosaccharides can be used for therapeutic purposes, right? Fucose, mannose, galactose, these have been used, for example, for CDG, disease-related studies. It's been used in cancer that they feed animals, and they've done clinical studies feeding these monosaccharides, and they've seen changes in outcome of the disease, especially if it's CDG outcomes. So it's important to find out these ratios and where is that exogenous monosaccharide going into to the cells, where is that ending up? So that was recently published in Journal of Cell Biology. You can look at the more details. It's an extensive study. They did a lot of work. We just did limited amount of work on figuring out exactly the location that the fucose is going to. This is another study with Dr. Lee. This is from Georgia State University. He's making an array of oligosaccharides. We've been talking about having standards for a lot of our experiments. And one limiting factor in glycobiology is having the right standards for doing the work that you guys are doing and we are doing. So he's tapped into being able to do chemoenzymatic synthesis of oligosaccharides, and he needed to confirm the structure to make sure that he's making the correct structure. So using MSMS, by going into MS3, we were able to basically confirm that the structure has a bisecting GlucNAc, and so this can be used by researchers building a library and maybe extending the bisecting GlucNAc and able to see whether he's been able to successfully extend the bisecting GlucNAc for that being used for external users as standards. So that has recently been also published. So another interesting study is, again, something recent we've gone into with Dr. Miller from University of Illinois. He was interested in to see how the sperm gets into the female tract, and how is the immune system, female's immune system, not basically rejecting the sperm. So Andel in 2007 had done a glycosylation study of sperms in humans. And at the time, she had figured out that Lewis structures were present in the sperm. And they postulated then that these Lewis structures could be responsible for the fact that basically the sperm can now be able to get into the tract and not be blocked by the female immune system. So Dr. Miller wanted to confirm this in porcine samples and see whether there's any additional factors, additional functional groups could be responsible for this. So we looked at the mass spec of N-glycans released from the sperm in porcine samples. And that is the full mass spec spectrum. We were able to see Lewis structures present in very small amount. The spectrum you see at the top is the extended version of the high molecular weight mass regions. And we see tiny amount, maybe few percent of Lewis structures. So Lewis structures are not significant portion in porcine. But bisecting GlucNAc was present significantly. So one feature that we saw in porcine is bisecting. So they also found bisecting in 2007. So definitely bisecting is postulated that bisecting and Lewis structures may be responsible for this. But they had also detected SIGLAC structures in the tract. And they wanted to know whether salicylic acid could also be playing a role in here. So we looked at the linkages of salicylic acids present in the O-linked. We did not detect any salicylic acid in the N-linked. They were expecting some N-linked, but we did not detect any basic cellulation in the N-linked. But the cellulation was present in the O-linked. And interestingly, we did MSMS, and we went to the MS5 in order to be able to do linkage analysis using permethylation and be able to distinguish between 2,3 and 2,6 and the ratio of the 2,3 to 2,6. And we found that the salicylic acid was determined to be a mixture of 2,3 and 2,6, but mainly 2,3 linked salicylic acids. So it is believed that SIGLAC are playing a part in here because of salicylic acids. So maybe salicylic acid, combination of salicylic acids, Lewis structures, and the bisecting GlucNAcs could be playing a role here. So another study is collaboration with Nicholas Heaton from Duke University. So he's studying influenza A and influenza B virus and trying to figure out a host system that could block the virus attaching to the cells, a host kind of antiviral therapy. So what he had done, they had done a wide GY study using CRISPR, and they had figured out that against influenza B. So they wanted to see basically what is popping up when they do a GWAS study against influenza B. What they found out was there is the beta-1,3 gluc-A transferase that is the only thing that was popping up. So this gluc-A transferase was the only thing that they showed that was unusual when they did the GWAS studies. They wanted to know what is the outcome of these gluc-A transferase, 1,3 gluc-A transferase. So they wanted to see whether having overexpression of these gluc-A transferase has any effect on glycosylation, N-link glycosylation at all or not. So when they did some lectin staining, and then we did the glycan analysis, we found out that the sialic acids are being replaced by glucose in the N-link glycans. So basically, it's outcompeting, the glucose is outcompeting with the sialic acids. And when you look at the way it's being made in the Golgi, actually the silyl transferases are at the end of the process where the glycans are being processed, whereas the glucose is present first. So they get added first, and then silyl transferases come later. So basically, it's replacing, the glucose is replacing the sialic acids. So that's why it's basically blocking the influenza B being attached to sialic acid, because there is no sialic acids, so that is being blocked. So they're going to possibly use this as tapping into this as a potential antiviral activity for all types of influenza that have sialic acids that they basically attach to sialic acids. So interestingly, that spectra is the normal spectra from the wild type, and the one that you see that is extended with very little sialic acids is the one that have glucose attached to the N-link glycans. Interestingly, there is a diagnostic mass that we see associated with the glucose containing oligosaccharides. They lose 32 mass units that because of permethylation, we can use this as a diagnostic mass to see that because you get a beta elimination of the uronic acid, you see this diagnostic mass, so we can use that as a diagnosis. So basically, with a drastic reduction in sialic acid moieties on the N-link glycans, influenza infection was drastically inhibited. And then we confirmed that with just running labeling the glycans and running that by HPLC and confirming that by mass spec also. We wanted to make sure that that beta elimination is not interfering with any of the assignments. So we did label and run the glucans, the glycans, and did MSMS to confirm those. And also confirmed the linkage of the remaining sialic acid. There was residual amount of sialic acid left after the introduction of the glucose into the N-linked pathway. And interestingly, the results indicate that the alpha 2,3 sialic acid linkage is marginally less abundant than the 2,6 from the remaining sialic acids that are still there. So these are just a couple of the projects that we've done in the last 6 to 7 months on various N-link glycosylation studies and O-link glycosylation studies. But what I'm excited about is being able to, moving forward, we're going to use FAMES. This is a modification of an ion mobility instrument, is an addition to the existing instrument. Basically, is an add-on, is an orthogonal method to LC-MS, so you can do LC and also use this ion mobility to guide ions. So I'm interested to use this for a lot of glycopeptide analysis and glycans to see how it separates and how it will help us separate some of these moieties, some of these isomers from each other. So basically what it does, it funnels in different structures. So you can start from basically giving it different voltages, and you can filter sialic acids, you can filter hexnax, and sialic acids, then different structures will pop up, so you get a different layer of separation in addition to LC. Because LC on its own may have limitations of just isomers coming out together, so if this is added on, maybe we can separate those isomers from each other. I think it's very powerful for glycopeptide analysis. I want to tell you about, we mentioned the courses from the Complex Carbohydrate Research Center yesterday. So for the last 30 years, we've done hands-on training courses in carbohydrate analysis. The last 2 years, they were virtual, but this year, again, they were in hands-on. So we will have them both virtual and hands-on in the future. The virtual one will be shorter, of course. These are hands-on courses. The first one is a polysaccharide course. So basically, you learn about composition analysis, linkage analysis. You have a module of NMR, molecular modeling, or mass spec. Basically, it's mostly on polysaccharide work. The second course is probably our most popular course, is release of N-linked and only glycans, mass spec analysis, HPAC analysis, and NMR, mass spec, and molecular modeling of glycoproteins and glycolipids. The third one is glycosaminoglycans, doing a lot of disaccharide analysis by SACS HPLC, by mass spec, and NMR studies. The fourth one this year, for some reason, was very popular because bioinformatics of glycoproteins, so a lot of glycoproteomics analysis, data analysis, looking at different softwares that are available for glycan analysis and glycoproteomics analysis. This is just for glycoproteins, so we don't cover any other glycoconjugates in this course. They're mostly in August, and the last course is in October. And with conclusion, I'd just like to thank our glycomics group. These are the folks which predominantly just do glycomics and glycoproteomics, and our collaborators. And we are fortunate with the funding because the funding from NIH, from Glycomip and DOE enables us to do these collaborations at no charge to other people. I'll be happy to take any questions.